All right, good morning. Thank you for the committee for accepting our late-breaking abstract here. These are my disclosures. This was also, like the last presentation, a collaboration of multiple institutions, 4 different institutions across Canada as part of this first-in-human study. So in the angio suite, we're used to getting beautiful images looking really at the lumen of blood vessels, but looking at the outside, whether it's using high definition fluoroscopy, looking at advanced reconstructed imaging, 4D imaging. That's all great. But what we really want to look at is what does the inside of the blood vessel look like? And I don't mean like this when you're looking at the inside during surgery, an open surgery, but what we really want to see is this kind of a childlike view of the inside of the blood vessel. This is a cartoon I watched as a kid. I did not have a sense that I would be doing something like this many years later, but this kind of a view looking at the inside of the blood vessel and flying as if you're on the inside. So we have that with something called angioscopy. And angioscopy has been available for decades now in the coronary vasculature. It's been available to go look at carotid plaques in the neck. However, we know that in order to bring something into the neurovascular space, we need improvements in flexibility, navigability, and improved safety. And so we have that now with a device called the MicroAngioscope, very similar in size based on a microcatheter platform. In a series of preclinical studies, we've tested this in cadaver models and a variety of animal models as well. And these are the images from some of those prior studies, looking here at a pipeline flow diverter placement in a rabbit model, where we've been able to assess wall apposition, thrombus formation, endothelialization. These are all very interesting, useful clinical endpoints that can guide clinical decision making. So the goal in this first-in-human study was to assess the feasibility and safety of the microangioscope as an adjunct in neurointerventional procedures. This is what the device looks like. It's based on a microcatheter platform. It has 2 ports on the back end. What we see here is this connects to a standard light source that's available in any interventional suite or OR. And then the other port, which is the smaller arrow, is connected to a endoscopy tower to provide high, up to 4K kind of imaging. In acquiring the images, we use a balloon guide catheter for temporary flow arrest and high flow irrigation. This clears out the blood, and you're clearing the view ahead of you, and then the microangioscope can be navigated anywhere where a microcatheter can go. These are our first 8 cases in patients. We started in the extracranial circulation. That was a case of a carotid web. The microangioscope was able to visualize and identify the carotid web in a way that was just not quite the same with standard 2D DSA. We subsequently moved to the venous side, and this was quite interesting. We did a case of a venous stent follow-up and subsequent stenting for stent-adjacent stenosis. I'll show you that case. Then it was very useful in flow diversion. Based on our preclinical studies, this was a very interesting use case, both for initial primary flow diverter deployment as well as in follow-up. And then another venous case and a web embolization. The microangioscope was safely put at target and deployed in all cases. So here's an example. This is a venous stent. In this case, the patient had a stent-adjacent stenosis, and you can see the initial stent at 6 months has been fully endothelialized. It's the first time we've really been able to visualize what's happening when we deploy these venous stents and what's going on in vivo. And then you can see after a new stent has been deployed, sorry if my marker's not shown there, the small arrow is showing the reflection against the fresh stent, and then the arrowheads are showing the previously endothelialized venous stent. Here's a video. As you go back and forth, you can kind of see in perspective what's going on there. That's a fresh stent that's been placed, and then what you see kind of ghosted there is an intimal layer over the prior stent. Here's another case. This is a dural venous sinus injury that was with a balloon and a small intimal injury that was not detected by DSA. It is detected here with the microangioscope. This is actually Vitor's case that he provided to us from St. Mike's. And then this provided us useful information that led to subsequent stenting. Here's my personal favorite. Again, this is a pipeline flow diverter. We were able to assess wall apposition, absence of thrombus, and successful deployment. And then in a subsequent case, you're able to see between those 2 arrowheads you do not see the metal of the stent anymore. We can tell this has been fully endothelialized, and this is a particularly useful clinical endpoint for subsequent antiplatelet management. So in conclusion, this is the first in-human direct endoluminal angioscopy in the neurovasculature. We were able to find that it was safe as an adjunct in these various neurointerventional procedures. We are seeing imaging both in cerebral arteries as well as veins, and we'll definitely need further evaluation testing in a broader cohort in different pathologies. We've been able to take this, thankfully, from the lab many years ago, and now in our first in-human case in the clinic. Thank you.